Because I don't know what else you.
Sorry.
Okay. So here would be kingdom, because that's the only one that makes sense there.
Mm, yeah. But before that, animal, plants, or fungus kingdoms. Yeah, that's correct. Okay. So
Okay.
the development of body plan in eukaryotic organisms is controlled by homeobox genes. These genes code for proteins that are able to bind to DNA, in turn specific genes on and off, and are known as transcription factors. So these proteins contain a sequence of base pairs that varies little between species within the animal, plant, or fungus kingdoms, right? That's what it is. Now you can move
Yeah.
for the next question, please.
Okay. Investigations into the activity of genes that control plans frequently use fruit flies and mice. One reason is that there are fewer public concerns. State two other reasons why fruit flies are chosen for research. Why
Hmm.
mice are chosen? They're, oh, we were talking about this. Mice are smaller, so there's less space needed to like store them.
Hmm.
So that could be one. Because so there's less space needed than the like
Hmm.
less like less expensive as well.
Okay, you just write the answer here, so then I'll explain it to you. Reasons why fruit flies are chosen for research into genes controlling the development of body plans are. We discussed about this last time. We said, like, as we said, the generation time, we said, like, genetics, the development is well understood of it. You just write the answer and then I will correct you here, okay? Just use your own mind and try to write the answer. Smaller light space. Mm-hmm.
Mm, I think that's it.
Mm-hmm. Okay, do we have the 4 mark? You have completed for the 4 marks or not?
No.
Smaller, less space, less expensive. So let me tell you about this. If you're talking about it, so we can say they are low cost, or we can say like they're easy to keep as well. This is for 1 mark that you can add here. And they have rapid reproduction rates that I discussed last time because they have the short lifespan, right? So any of the organism that has a short lifespan, so it's easy for us to observe the changes in it, whatever the modifications that we have made with it. So in a similar way, fruit fly genetics is well understood because we are using this model organism since long, right? So it means like that's quite easy for us to understand any of the modifications that we are making inside of it, so we can easily understand what would be its expression, right? So they have simple genetics actually, just a simple body plan as well. So by keeping all these points in mind, so we can say like we are using this organism, in research into genes that's controlling the development of the body plan, right? So that's why we are using it. They have many mutation structures that are observed with the light or low powered microscope. So you don't need too much like high resolution microscope for this. Any of the change that you can easily observe in the Drosophila, right? So that's why this fruit fly, the Drosophila, is used in research into genetics, right? That's controlling the development of the body plans. Are you getting this point? Could you please just
Yeah.
rephrase your answer again to write it properly for 4 marks?
Okay.
Thank you.
I'll just type it then.
Yeah, you can type it. It's not typing.
Just wait, me to press the typing button.
Hmm. We are dealing with fruit flies, not mice.
Yeah. Okay, so that's that one.
Fruit flies are easier to keep, so they have low cost. They also reproduce faster, and they have the shorter lifespan. Do we have all of the this is for 2 mark question, right?
Yeah.
It's
So then low
fine. So,
cost, reproduce faster and shorter
okay,
lifespan.
okay, okay, that's fine. So now you can move for the next question, please.
Okay. Just reasons why mice are chosen.
Hmm.
Mm,
Again,
is
there
it
would
not
be
the
of the
same?
same
Yeah.
sort of answer we can say. The one important thing is mice are more similar to humans, right? And
Okay, yeah.
as per we are saying,
As in like comparison to fruit flies.
yeah. So the effects seen in the mice are, generalizable to more than one species. For example, any of the change that we can observe in mice, so we can generalize that change in all different organisms that's related to the mammals, right? Specific to the mice. So they are low cost, cheap, easy to keep as usual. They have the short lifespan. These are some common, you can say, characteristics or the properties of the model organism that we commonly use for. All right. So that's how you can add this here. So using more than one species is needed to demonstrate conservation of the base sequence that we have observed, like few of the base sequences were conserved across the different species that we worked on.
Okay.
Yes, have you done?
Yeah.
So why you don't move for the next question, please? Move for the next. This will be the question. Please read out the question and let me know what exactly the focus idea that you understood from here.
Marfan syndrome is a genetic disorder that affects the connective tissue. It's caused by mutations in the gene known as fibrillin 1. Figure 1 shows the effects of two different mutations that can occur in the gene. Identify the type of gene mutation shown in mutation 2 in figure 1. That's the original. Oh, this one's substitution.
Identify the type of gene mutation and justify it, how it's mutation.
This one was the substitution one because the C is being substituted for the U.
Yeah, it's a substitution mutation. You're right, right? So the second part of it, amino acid P is, yeah, yes, beta.
I'm just writing down the answer. Okay.
This is for 2. So the cytosine has been
Yeah.
replaced by yourself. No, the question is show the mRNA codon that codes for the 4 different amino acids.
Yeah,
Yeah.
it's for the first question.
Okay.
The table one shows the mRNA codon that codes for different amino acids. Okay.
Mm-hmm.
Okay. Use the information in table one to identify amino acid P from figure 1. Use the information in table 1 to identify amino acid P from figure 1. Okay. So it's original UUC. So that's UUC. So isn't it 5?
Phenylalanine. It's
Yeah.
phenylalanine, right?
Yeah.
So the P is for phenylalanine. Okay.
Yep.
Next question.
We see information in Figure 1 and Table 1 to explain the effects of the following on fibrin proteins for which the section of DNA codes.
What's the focus idea? Use the information in Figure 1 and Table 1 to explain the effects of the following on the fibrillin protein for which this section of the DNA codes. You have mutation 1, mutation 2.
Yeah.
So you can see here the amino acid triosine that would replace cysteine.
Okay. So what do I just have to say? It would how it would change,
I'm
like the
talking the mutation 1 would
structure.
affect the fibrillin proteins, right? That's what I'm saying. The
So
mutation
then
1
would I
would
just
affect.
talk about how it affects protein structure if that's what's changing?
Yeah. So disulfide bond formation would be altered here.
Oh, okay.
So if the disulfide bond would be affected or altered, so ultimately the tertiary structure of the protein would be altered. That's leading to the non-functional protein. That's possible because disulfide bridges are formed in the tertiary structure, right? So in mutation 1, that would affect the fibrillin protein in this way, right? So one amino acid being replaced with another can influence the way that the protein structure develops, that we know about it. So disulfide bonds form between cysteine amino acids. So the removal of cysteine will influence the folding of the protein, right? So because in cysteine we have the disulfide bridges would form. So the end result of this will be the altered tertiary structure and potentially non-functional protein.
Okay. So I would have to say that the changing.
You
No,
should
so I
remember here one thing, like there you need to explain the effects of the protein, right? So it means examiner would look into this in terms of what would be the effect of the protein, right?
yeah. Wait, you were saying something about removing
Uh,
something that would change the folding.
in mutation 1,
Yeah.
I
You
said
said remove.
amino acid tyrosine would replace cysteine.
Okay.
Yeah. And this is what you will see in both of these structures. The tyrosine would replace the cysteine, and cysteine was actually there that was responsible for the disulfide bridges there because the sulfur was present in the cysteine, right? So if the tyrosine would replace the cysteine, so automatically it would affect the disulfide formation, right? And if the disulfide formation is affected, so automatically that's going to change the tertiary structure of the protein, right? That's leading to the non-functional protein. Are you getting this point?
Yes.
Okay, now you can write it down, please.
Okay. So is that fine for an answer? Is that fine?
Okay, okay. Disulfide bond would be altered, so the tertiary structure will also be changed as tyrosine replaces cysteine. Yeah. Now you can move for the mutation 2, please. What is in mutation 2? Mutation 2 would affect the fibrillin protein. How it would be, that's what you need to talk about.
So mutation 2.
Look into the mutation 2 there.
GG. Nothing's changed.
Nothing is changed. Means no change to the amino acid sequence, right?
Yeah, so it will still code
So
called
tertiary of the 3D
5.
structure of the protein will remain same here, right?
Yeah.
And it would be unaffected or unaltered because if there would not be any change, apparently that we can see in the mutation 2, right? So no
Yeah.
change to the amino acid sequence. because the first amino acid in the sequence that you can see that still be the phenylalanine, right?
Yeah. Okay. So is that fine? Click on the next question.
Yes, please. Read the question first and try to mention here the focus ID and then start answering.
Figure 2 below shows a third possible mutation that could occur in the same section of the gene. Explain how the type of mutation demonstrated by mutation 3 can have a greater impact on protein function than the mutations shown in figure 1. What's figure 1? Okay.
Explain how the type of mutation demonstrated by mutation 3 can have a greater impact on the protein function than the mutation shown in figure 1.
Well, mutation 3, you've gotten rid of one of the bases because it's shorter now.
Mutation 3 is a deletion mutation. Can you say?
Yeah, because they've deleted one of the bases.
So when mutation 3 is, if it's a deletion mutation, means the second base is deleted from the sequence. Is it?
Yeah.
So this results in the frame shift, right?
Yeah.
So then we can say every codon downstream from the mutation is altered definitely, because if this deletion, so rest of all means downstream codons would be changed, would be altered, because they have to go for the triplet, because one is removed from there, so now the next one will become to part of the triplet, and this is how all the downstream codons will be changed. So if
Okay.
the mutation occurs early on this sequence, then many amino acids can be changed, right?
Yeah.
That's what it is. So the protein can be shortened or lengthened if a stop codon is created or removed.
Okay.
That's how we can answer this question, right? So deletion is actually, we have discussed very clearly in the topic when I was talking about this specific type of mutation. When any sequence in specific base is getting removed from there. So as a result of this, the all downstream codons would be changed, because they have to keep themselves in a triplet form. Like if 3 were there, 1 was removed, right? The
Yeah.
second was removed. So it means now because two bases would not code for any amino acid, right? So they have to become 3. So for this purpose, the second one that was deleted, in order to compensate this, the next downstream codon will come to attach with this to become the triplet. So this is how this change would lead up to the end of the codons towards the downstream, right? Are
So
you
I'd say
getting some? Is it making some sense or not?
it is. So I say that the mutation type is deletion. So that
Yeah,
means the all
that's
the
actually that's results in the frame shift,
okay and
right?
okay. Yeah. And then the frame shift means that the codon sequence is changed, like altered.
Actually, this is what we are saying. The frame shift mutation is actually when the deletion of the nucleotide bases, right? Um, in numbers that are not multiples of 3, right? Means
Yeah.
that's not of 3 that's going to be deleted, only one is. In frame shift mutation, for example, we said that one base is deleted from there. When one or more of the bases are inserted or deleted, this is what we say. So now bases are read in group of 3, is it?
Yeah.
So their addition or deletion in any other than means then multiples of the 3 disrupts the reading frame sequence. The next reading frame sequence would be completely changed, right? So the concept of frame shift should be clear to you, right?
Yes.
So like if I can show you this here, so you will see this exact change here. I'm just let me put it for you to understand how the frame shift would affect it. Um, if it's allowing me to add up here. So for instance, I'm just adding it for you. Yeah, it's here. Thanks. It's fine. Look at this. Can you see it? Just
Yeah.
look at this here. What's going on? For instance, at this point, there's a deletion or something is like this one. This is changed here, right? So
Yeah.
see how the deletion of the nucleotide CG resulted in an altered amino acid sequence. You can see it was for arginine, right? At this
Yeah.
point. But the fifth amino acid arginine is not now translated as glutamate. How this is possible? Just for the reason because the C was deleted from here, suppose. This CG that you said, this was codon 5, right? So now
Yeah.
codon 5 is here, that's of GAG.
Okay.
And here codon 5 was CGG, right? So now the
Right.
codon is changed because I said the downstream codons would all change. This is what we are saying, like it's a frame shift, right? Are you getting this point?
Yeah.
Now you can write the answer, please. Done?
Yeah.
The mutation type is deletion, and that results in the frame shift, and every codon bound downstream from the mutation is altered. So this will then change the type of amino acid produced. Yeah, that's good. Perfect. Move for the next, please.
Okay. New mutations have been discovered by this in the gene, and it's thought that mutagens in the environment may have contributed to the occurrence of these mutations. Suggest two examples of wait, of mutagens that could have caused these mutations. X-rays is always one of them. Like those always can cause mutations.
Okay, just write down the answer. Then I'll see and I will rephrase it for you to help you to correct this. Use your own mind first, please.
Mm.
Hmm. Do you want help here?
Yeah, I can't think of a second one.
In one, you can see here. New mutations have been discovered in the FBN1 gene, and it is thought that mutagens in the environment may have contributed to the occurrence of these mutations. So just two examples of the mutagens that could have caused these mutations. That's quite simple. Yeah, take any
Huh?
two or three.
I can't
White
think of
cells,
this.
bacteria, X-rays, like any, in any of them that you can take in. The X-rays, as you said, UV rays, ionizing radiations, even
Yeah.
deaminating agents would change. Base analogs are changed. Alkylating agents are there, methyl, ethyl groups. That means a lot of examples we can take in. Viruses, bacteria, so you can take them. So means and a lot of other examples of the mutagens that you can add here.
Okay.
Getting correct?
Yeah.
Okay, please move for the next question. Yeah, it's here. We are here on 3A now. Read out the question, please, first, and let me know what's the focus idea, and then we will discuss.
Okay. Figure 1 shows the growth of an E. coli bacteria population that is cultured in a medium containing both glucose and lactose. Between points P to Q in figure 1, the E. coli cells are used in glucose, and between points R to S, the cells are using lactose. Describe the growth of the E. coli population over 10 hours. Okay.
So what are you getting from here?
So looking at the graph.
Describe the growth of the E. coli population over 10 hours. Look into this graph and what it is giving it some idea about it.
Yeah. Um,
Just explain it to me, please.
I'm trying to see, like, is it 0.2? 0.
Mm-hmm.
I think it looks like 0.2. So from 0.2 all the way up to like, it's not
Mm-hmm.
exactly at 2, so like 1.9-ish.
Mm-hmm. Okay.
So from 0.2 to 0.9, the bacteria like population increases.
Okay.
Wait, let me write that down.
So we just need to describe the growth of E. coli population over 10
Yeah.
hours from between PQ and RS, right?
Yep. So it increases. So between PQ, can you say that between PQ and RS there's a steady increase?
You can look into this given graph that's here for you. You can see the bacterial growth increases from log 0.2 to log 0.19, 1.9, sorry, over 10 hours. That's what you can
Yeah.
see here, right? Like if you look into this, okay,
Yeah.
0.2 to log 1.9 means 1.9 up to this point, right? So if you just go from this to 10 hours, this is 10 hours. If I can, you can see it like it's getting increased, right? In all of
Yeah.
this, what you can understand here, right? So there is an hour of no population growth as well between 0 hour and 1 hour, right? The population growth increases slowly between hours 0 to 1, right? So the population growth stops or levels off between hours 4 and 5, or QR you can say. QR. Can you see the QR here? It's level off here, right? So the
Yeah.
population increases more quickly between 1 to 4 hours, means PQ, than between 6 to 10 hours. Can you say it?
Yeah.
Is this not getting increased more?
Yes, it is.
So data description, you can see like sometimes it often appears very straightforward, but you should remember this can be trickier to gain all the available marks that like that you can say like then it might seem. So use numbers from the data to back up your point, right? Whenever you are I mean attempting this question, so use the reference points from there, right? That's very important, right? And
Yeah.
yeah, don't need to go for the explanation if the question that you have like that, if that's not the requirement of the question. So in this given question here, so you can simply say like we just need to look into this, describe the growth of the E. coli population over 10 hours. The graph is in front of us. So we can simply say the bacterial population increases from log 0.2 to log 1.9 over 10 hours. 0.2, it's almost 0.2, right? It's going up to, you can say, up to 1.9 hours. Is
Okay.
it? This is
Yeah.
the, because this graph is complete, I mean, that's very evident, and it's showing us like how it is getting increased from 0.2 to 1.9. Okay.
Okay.
So just write down the answer, please, now.
I think I did. I wrote down like 3 different points.
From 0.2 to 1.9, the bacteria population increases. Between PQ and RS, there is a steady increase. Between QR, there is no E. coli growth, means the levels of, like we can see here. Yeah, that's fine. This is how we can mention it. Take the points from there. You can, like, you should also mention the time here as well. Means not only PQR, but you should mention the value from there. What was the, you can say, the time actually, right? So there is an hour of no population growth between 0 hours and 1 hour, right?
Yeah.
Can you see any growth here, the 0 and to 1?
Yeah.
There's no growth from 0 to 1. It
It just
started from
kind
2. 0 to 1, and from 1 to 2 then it started, right? 0 to 1, there's no growth. Like you can see
I
here
thought
the
you meant
graph,
from like the hours. I was like, there is growth from there, but okay.
like this one, right? This is
Yeah.
that you can see like there's no growth here. Okay. So
Mm,
if there's no growth at this point, the population growth increases slowly between over 0 to 1. It's no growth and very slow growth, right, at this point. Because we can see here, like up to this upward, like bar is, line is showing us like now this is getting increased.
Okay.
Okay.
Yeah.
So the population growth stops at the levels of between over 4 to 5. Is it? Just
Yeah.
get to the 4. This is 4, and up to this point this would be of almost 5, because it's 4 to 6, so 5 is here, right? 4 to 5 we can say it's level of.
Okay.
So the population increases more quickly between 1 to 4 over PQ than between 6 to 10. So you can compare it, 6 to 10, and the first one is 2 blocks which are formed here. So we can say the population increases more quickly between 1 to 4 hours than between 6 to 10 hours. Is it?
Yeah.
How we can analyze this? This straight upward move, like this line up is getting up like this.
Okay.
This same sort of graph we can't get here. It's getting bit like this. It's showing us like the growth rate was here more as compared to this point between between RS. Means
Yeah.
PQ is showing us more growth as compared to RS. This graph is telling you about this. Okay.
Okay.
So, but keep in mind, take references again and again from here, and mention the time and as well as log of E. coli growth, right? So that's how you can get the good score here in the graphical representation questions. Okay.
Okay.
Yeah, move for the next, please. This one. So just an explanation for the trend in bacterial growth seen between points QR in figure 1.
An
Yes,
explanation. Okay. Between Q and R.
write down the answer. Now I've already explained this everything there, right? So now you just need to write the answer from here.
Okay.
So just an explanation for the trend in bacterial growth seen between point QR in figure 1.
Okay.
Mm-hmm. What do you think? What exactly it would be?
Um,
What? First, try to find out what's going on in QR, in
well,
between
the
QR.
bacteria growth is stopped for a little bit, so it's temporary. So I've already
Q
written that the
in
bacteria
QR.
growth.
Now the question is why? What do you think? Why this situation was created?
I was going to say that, like, obviously
What?
the bacteria was feeding on the glucose and the lactose. So maybe at point QR, they've both, well, they both couldn't have finished.
Not both actually.
One of them.
Primarily bacterial growth. I mean, the bacteria they were using the glucose, right? Once it is used up, it's finished. So
Yeah.
it takes time to I mean, to synthesize the new enzymes for the lactose, right? During
Okay.
that period means until the new enzymes are synthesized for the lactose. So there's a QR, like where there is no growth, right?
Okay.
So that's why we can say like glucose is running out, so bacteria need to switch to using lactose, right? So it takes time
That's
to synthesize enzymes that's needed to break down the lactose there instead of glucose, right?
needed. Yeah.
So lactose binds to the repressor protein and preventing it from binding to the operator. This is what we have already discussed in the lack of prawn, right? So
How do
lactose,
we how do we know that glucose is the one that finishes first?
yes,
Or it's just like,
that's you need to think about it. That's
okay.
that's that's what you need to get this from the given like this question here. That's we have to understand because the question is asking us below you shows the growth of the E. coli bacteria population that is cultured in a medium containing both glucose and lactose. And this is in the lac operon we studied. If the glucose is finished, so then the lactose would be utilized. In the presence of glucose, the lactose will not be there to be utilized. This is what we have studied in lack of prawn. You can get back to that specific topic and you will see both were present in that medium, but the bacteria was using the glucose first, right? Until the glucose is there, so lactose was not, the enzymes related to the lactose were not synthesized, right? Once the glucose finished, means that the glucose is absent now. Once used up means glucose is absent, right? So at that time, so the lactose is there to be now synthesized. This is the time when because at this point you have to, this is actually the application of the lack of prawn question, right? So that's what you need to understand here, because between point q or on the graph you can see the growth curve levels off. That's showing that bacterial cell division has temporarily stopped, right?
Okay.
So you can see here in the path A, we talk about this, that states that the bacteria are making the switch from glucose to lactose as their food source at this point, right?
Okay.
So you should, means like reason that your knowledge of the lac operon will help you here, because lac operon that we have studied there, and you once you will revise it again, so you will see. So E. coli prefer to use glucose as their food source, remember, and do not expend energy synthesizing enzymes that they don't need. Why they need to go to synthesize the lactase if they already have the glucose there, right? That's the waste of energy. They don't want to expend that energy in this way. So this means that they are unable to make an I mean instant switch to the lactose means a breakdown once the glucose food that's run out, right? So
Okay.
so this is what you should keep in mind. So they do not yet have the necessary enzymes there, right? So there is therefore the temporary pause in the growth curve. Are you getting this point? So it takes
Yes.
time to activate the genes that code for the enzymes that break down lactose. So first lactose binds to the repressor protein, preventing it from binding to the operator region. This enables the RNA polymerase to bind to the promoter region and initiate transcription. And this enables RNA polymerase to bind to the promoter region and initiate the transcription. So once this sequence of events has occurred, the enzyme for lactose digestion that's produced, and the bacteria can use the lactose now as a food to fuel its growth, just like the glucose that it has used before, because that was its preference, right? So now that was used up, so it's the compulsion for the bacteria to turns or to switch to the lactose now. But definitely the transcription and translation is not something just to click on the button and it would be produced. No, it takes time, right? So
Yeah.
that's what we should keep in mind. So now you can just explain this why in this way. Between QR, the growth of the bacteria temporarily stops due to the glucose running out. So the bacteria needs time to synthesize the enzymes needed to break down lactose. Perfect. Move for the next please. This question.
Yeah. So the E. coli bacteria needs to synthesize the amino acid tryptophan
Tryptophan. It's a tryptophan.
in order to build proteins inside their cells. Tryptophan synthesis requires enzymes that are coded by 5 structural genes. The synthesis of tryptophan is controlled by a region known as the TRP operon.
Tripopran. We talk about this as well. Tripopran, the action of which is shown here in figure 2 below. In the absence of tryptophan, when the tryptophan is present. In both of the cases, we can see.
Okay, so just how it controls the synthesis of it. Repressor. So we have a repressor protein. So it means that if you have a repressor protein, wouldn't it have to bind to the repressor protein? So how would that control it? Well, if it binds to it,
we talk about this. If you see in the because in the presence of tryptophan and the absence of tryptophan, if you talk about in the presence of tryptophan, so tryptophan binds to the repressor protein, right? So the repressor protein binds to the region Z. That's actually the operator region, right? Region Z, right? That's what you have again get back to the tryptophan control that we talked about before. So enzyme X, you can say the RNA polymerase is prevented from binding because the repressor protein binds to the region Z. So transcription cannot occur.
Yeah.
Right? This is what I'm talking about in the presence of tryptophan.
So the RNA is prevented from binding.
RNA polymerase is prevented from binding. Why? Because the repressor protein binds to the region Z, the operator region first. So no transcription is possible here.
Okay, so
getting this point? This is in the presence of tryptophan. Tryptophan binds to the repressor protein. So repressor protein binds to the region Z or the operator region. So enzyme X means RNA polymerase. Enzyme X is actually the RNA polymerase. That's prevented from binding. So the transcription cannot occur. Are you getting this point?
Yeah. So then the next one suggests the benefit to the E. coli of controlling tryptophan synthesis in this way. Okay. Suggest the benefits.
You are not talking about in the absence of tryptophan. Why you're not mentioning? Because in both of these cases, we need to talk about it. We have to talk about in the absence of tryptophan as well. This is what you have explained in the presence of tryptophan. But what would be the condition in the absence of tryptophan?
In the absence, it wouldn't bind to the repressor protein.
Yeah. So the repressor protein is unable to bind to the region Z. And ultimately, RNA polymerase can bind easily and the transcription occurs. Right? So this is what the opposite to that was in the presence of.
So this one transcription will occur.
Yes, definitely. The tryptophan doesn't bind to the repressor protein, so the repressor protein is unable to bind to the region Z or the operator region, right? So enzyme X or RNA polymerase binds, so the transcription occurs. Context should be clear, remember, whether tryptophan is present or absent. This context should be clear there. Means whether you're talking about in the absence of tryptophan or the presence of tryptophan. That's your examiner will give you marks on this, okay? Keep in mind.
Yeah. Suggest the benefits to the E. coli of controlling tryptophan synthesis in this way. I'm not sure.
You're not sure about it?
Yeah.
Okay, just listen. The very simple and straight thing is tryptophan production stops when when the tryptophan is present, right?
Yeah.
If the tryptophan is already present, so if the production would not be stopped, so definitely the tryptophan would accumulate there. So
Yeah.
it means like when tryptophan is not needed there, but still you are there to synthesize the tryptophan. It would causes accumulation and as well as definitely it's the waste of energy. So energy is wasted in this way. So that's why so tryptophan production stops when it is present, right? Or once it is accumulated. So when no more the tryptophan is needed, so automatically energy is not wasted.
Okay.
Right? Are you getting this point? So
Yeah.
tryptophan production is stopped only when the tryptophan is present there, so that more tryptophan would not synthesize and that would not accumulate there, so that would not actually there to waste the energy, right? So this is
Yeah.
what actually the tryptophan regulates its own production, right? So once the plenty has been produced and levels of the tryptophan inside the cell rise, it binds to the repressor protein, enabling it to bind to the operator and preventing RNA polymerase from binding to the promoter, right? So this means that the bacteria stops synthesizing tryptophan, and this is how it's saving energy.
Okay.
All right, Corinne?
Yeah.
That's great. So write it down, right? Write the answer there. Done?
No.
So this is how tryptophan control its own production.
So we said that the tryptophan production stops when
When it is present in the plenty, or its production is rises in the cell. For that purpose, the tryptophan get back to bind to the where it would bind
1 second. To the repressor protein.
in the presence of, we said, like it would bind to the repressor protein,
Yeah.
right? So the repressor protein binds to the region Z, the operator region. So enzyme RNA polymerase is prevented to bind from there because the repressor protein binds to the region or the operator region. That will stop the binding of the RNA polymerase. It would actually save the energy. If the tryptophan is not needed, so why it should synthesize? Okay, are you getting this point or not?
Save energy. And we said that it can regulate its own production.
The tryptophan production stops when its production rises in the cell, and it saves energy. So tryptophan regulates its own production so that it would not accumulate or something. You can add it here, like you can mention this as well, like the tryptophan will bind to the repressor protein, so that would bind to the we discussed about the operator region, right? So once the repressor protein binds to the operator region, so RNA polymerase is prevented from binding.
Bind to the operator region.
Yeah. So no tryptophan production would be possible there.
Yeah. So if it binds to the operator,
just write it down by yourself. Why you not just write down by yourself now? Whatever I have explained to you, try to rephrase as by using your mind and complete the 4 points say for the 4 marks. Yes, couldn't be done. That's done.
Yeah.
Okay, great, wonderful. That's fine. It's fine. So the tryptophan production stops when its production rises in the cell, so it saves energy. Huh? Stops when its production. Okay, it saves energy. Okay, tryptophan regulates its production and it binds to the repressor protein, allowing it to bind to the operator region and stops the RNA polymerase from binding to the promoter region. Okay, that's fine. Next one, please. Comment this question.
Okay.
4A.
Hormone estrogen can diffuse into the human body cells. Once inside a cell, estrogen diffuses through the nuclear pores into the nucleus, where it can influence the process of transcription. Figure 1 shows the actions of the estrogen. Okay. Explain how ER acts as a transcription factor in figure 1. So the estrogen receptor. So how does it act as a transcription factor?
So,
Well, it will bind definitely. So the estrogen will bind to the estrogen receptor.
write down the answer here. Whatever you're getting, just based on that, based on your understanding, try to write here. As per according to marks, right? Then we'll see how we can improve it further. How many marks we have for this question?
Um,
It's not mentioned here, right?
not like
It
the
is.
page.
Yeah. 3 mark question. It's a 3 mark question.
Okay.
Yeah, 3 mark question here. Please draw here up so that we can complete this worksheet.
Well, it's only 2 marks, right? So is this enough for two
It's,
marks? Or do I have to?
it's 2 or 3. We have 3 marks for this question.
Oh, is it?
Yeah, 3 marks.
Okay. All right, well then.
Explain how ER acts as a transcription factor. So the hormone estrogen can diffuse into the human body cells. Once inside a cell, estrogen diffuses through the nuclear pores into the nucleus, where it can influence the process of transcription. So figure 1 shows the action of the estrogen. So this is estrogen here. So this is estrogen receptor. So then you have the cofactor and the DNA and the transcription occurs. So here we
Yeah.
can see the estrogen receptor, how it acts as a transcription factor. So we can say estrogen binds to the, estrogen receptor. So it causes some structural change, right? So estrogen receptor can bind to the cofactor. So cofactor can then bind to the promoter region, right?
Okay.
So on the DNA, definitely promoter region would be on the DNA. So RNA polymerase can bind to the DNA and initiate transcription. So this is how we can say.
Okay.
Actually, you just need to apply your concept or your knowledge of the transcription factors to the sequence of events that's shown in this given figure, right? So the sequence may be slightly more complicated than like we have here, but we just need to remember the action of the transcription factors is, I can say, all about influencing the ability of RNA polymerase to bind, right? Because the transcription factor is influencing the ability of the RNA polymerase to bind to the start of the gene, actually, right?
Right.
And definitely here this is enabled by shape changes in the series of molecules that are initiated by the binding of the estrogen to ER, right? So actually this is the configurational changes which would occur here. So this is how you would answer this question. Any of the 3 points that you can write, you can say estrogen binds to the estrogen receptor and causes the structural, the shape change, right? So estrogen receptor can bind to the cofactor. So cofactor then bind to the promoter region, and RNA polymerase can bind to the DNA, right? And initiate transcription.
Okay.
Write down the answer, please.
I did.
Which is on the DNA. Yeah.
Okay, so in certain types of
So,
breast cancer.
so actually, it initiates the transcription. You can mention this, which is on the DNA, and it
Oh,
initiates the transcription. Are you getting this point?
yeah.
Great. Move to this question, please.
In certain types of breast cancer, the process described in Part A may lead to the development of estrogen-induced tumors. Ta tamo Tamoxifen, one of the first effective treatments for these types of cancers, and is still used with great success. Suggest how it's an effective treatment for breast cancer.
Estrogen receptor, cofactor, yeah. So just how the tamoxifen is an effective treatment for the breast cancer.
Mm. Well, okay, should I just write down the answer?
Definitely, you need to write the answer now.
The shape of the tamoxifen isn't a complementary to the cofactor, is it? Like that's what the thing is showing me, right?
Actually, it binds to the estrogen receptor, right? So it prevents estrogen from binding to the estrogen receptor. That's what it is. So tamoxifen is not complementary in shape to the cofactor, if you can say it, right? So estrogen receptor cannot bind to the cofactor. So automatically, if the estrogen receptor can't bind to the cofactor, so no transcription can happen.
Okay.
You don't need to go for the tamoxifen, like what it is or how we can. That's not our concern. You should be familiar with the transcription factors, right?
Yeah.
And that's in the figure 2. That provides you with all the additional information that you need to answer, right?
Yeah.
So you can see the figure 2 shows that this tamoxifen binds to the ER, right? That a slight difference in shape between the tamoxifen. And
Yeah.
estrogen prevents like this ER estrogen receptor from binding to the cofactor, right? So this will prevent the cofactor from binding to the promoter, and RNA polymerase will not bind, and the gene will not be transcribed, right? So that's what it is. Are you getting?
Yeah.
Okay, good.
Okay, so
Now you can
estrogen,
move for the next question, please.
yeah, estrogen receptor is said to act as an activator in the process of gene expression, but some transcription factors prevent genes from being expressed. So just two ways in which transcription factors might prevent a gene from being expressed. What would prevent a gene from being expressed? In transcription factors. Transcription, so RNA. So RNA.
So just two ways in which transcription factors might prevent the gene from being expressed.
Yeah. If the RNA polymerase doesn't bind to DNA, wouldn't that stop the gene from being expressed? I think that was one of our
Yes,
one
definitely.
we said.
Yeah, that's one of the points. One more point that you need here. One more point that you need here. One is the
One.
preventing the attachment and the binding of the RNA polymerase to DNA. That's what we know, right?
Yeah.
We can also mention here like preventing the movement or the progress of RNA polymerase along a gene,
Okay.
right? That's what you can mention as well. Just preventing the movement or the progress of RNA polymerase along a gene.
Yeah. So that would give us the 2 marks.
Yes, that's fine for 2 mark question. Yeah, we just need to mention two of the points here. The next question is transcription factors regulate gene expression at the transcription stage. Describe two regulatory mechanisms that control gene expression at the post-transcriptional level.
Oh, one was the one we kept talking about. It was like the genes slicing.
Post-transcriptional level. We talked about this, yeah. Splicing, yeah.
Slicing. Okay.
And one because splicing is what actually? What we are doing in the
Cutting.
splicing? We are just removing the introns, right? And just fusing together of the exons. That's what we have already talked about this, right? So like this is one part of it, regulatory mechanism that control the gene expression at the post-transcriptional level. We said the splicing. Splicing means like removal of the introns and fusing together the exons. That's how I explained to you. One more point that you can write it, how many 2 marks question here. So yeah, that's what it is.
Removing the introns and
Splicing, you should use the term first.
Yeah,
Splicing
so genes.
itself. Gene splicing, this is actually the further description of the splicing, right?
Okay.
Splicing means, what does the splicing means? It means like we are removing the introns and just fusing the exons with each other.
Okay.
Are you getting this?
Yeah.
Great. All right.
Yeah.
So now you can move for the next question, please.
I don't think there's another question. Oh, there it is. Fine.
5A. Read out the
A
question,
mutation
please.
in the HOX13 gene has been associated in some studies with the limb condition known as syndacty. Whatever.
Syndactyly. This is syndactyly, beta.
Okay.
Conan.
Define the
This is
term.
syndactyly means like the you would say the fingers would be like joined with each other. So define the term mutation. Suggest likely role of the gene HOX13. Mutation is simple.
Mutation, a
Likely
change.
role of the gene HOXD13.
Okay. Suggest a likely role of the gene. Mm.
What do you think? What should be the role of it?
It would be.
Is this not like the this Hawks D13 is just defining the position of the body parts during development?
Okay.
Or
Yeah.
we can say, is this not Hawks? Because this is syndactyly, means the web between the fingers is not removed, means apoptosis is not occurred. We can also say like it means the Hawks D13 is regulating the apoptosis.
Okay. Yeah.
Are you getting this? Is this giving you some sense or not?
Yeah. Okay.
Okay, get it. Great. So these 2 points that you need to mention here.
Okay.
Next, please.
Should I write it down really quickly?
Okay. Yeah, write it down, definitely.
The HOX gene and apoptosis, right? Regulates mitosis and
Yeah, simply
apoptosis.
you can say the HOX13, HOX D13 is there to regulate the apoptosis.
Okay. We suggest, okay, wait, the gene, something codes for the protein. This protein can bind to the promoter of another gene and initiate its expression. Suggest how it could initiate the expression of this. Well, it's always with the RNA polymerase, so it helps the RNA
It
polymerase
means
bind
HOX
to the
D13 is
DNA.
a transcription factor, what I can see here.
Okay.
Is this not playing? Because HOX13 could initiate expression of EPH7. Definitely, it looks like it is a transcription factor. It actually enables the RNA polymerase to bind to the DNA. So RNA polymerase initiates the transcription, right? What do you think about it?
Yes.
HOX13 is a protein, right? So,
Yeah.
so that it functions by binding to the promoter region. So this should be an immediate like giveaway. We can say that the protein function as a transcription factor. It's transcription factor that initiates the gene expression. Do so by enabling the action of the RNA polymerase, the enzyme that carries out transcription actually. Are you getting some sense from this?
Yeah.
So HOX D13 is the protein that's acting as a transcription factor. So it enables the RNA polymerase to bind to the DNA at the start of the gene, and RNA polymerase initiates the transcription itself. That's what it is.
Okay. Yeah,
Yeah.
got it. C. A mutation in the HOXD13 gene can lead to a non-functional protein. Explain how a mutation can lead to the synthesis of a non-functional protein. So that's always okay. So if it's a non-functional protein, it means there's been a change in the sequence of amino acids.
Yeah, definitely. There is a change in the amino acid sequence.
And if that happens, then the like structure will change.
Yeah, exactly. This would change the tertiary structure. I mean, different bonds form between the amino acids, right? If the change in the amino acid sequence, automatically that would change the bonding and all that pattern. So the protein folds differently, right? If the sequence is changed, so protein folds differently. It means like it altered the tertiary structure of it, right? So the protein is no longer complementary to the promoter. That's start of the EPHA7 gene, right? So it cannot bind to it to initiate the transcription because that's playing its role as a transcription factor. If the protein folding is changed, so automatically it's not going to bind to the EPHA7 gene and it would not initiate its expression there because it's quite changed now.
So it
It makes
can't
sense?
so it can't initiate transcription.
Transcription. Yeah. Because now the protein is changed. Because we know like the Hox D13 is playing its role as a transcription factor. Explain how the mutation can lead to the synthesis of non-functional Hox D13 protein. So we are saying the mutation can lead to the synthesis of non-functional HOX13 protein. So there's a change in the amino acid sequence. So different bonds form between the amino acids. So the protein folds differently and ultimately alter the tertiary structure. So the protein is no longer complementary to the promoter and it would not be able to start the EPHA gene. So it cannot bind to its initiate just to initiate this. Just move for the next, please. Next
Okay.
question.
Um,
Yeah.
okay.
You have the
I would
question,
say
please?
it was apoptosis, and then it's because the
Yeah.
tissue in
Yes.
between both of the fingers,
Yes. Because
the
this is apoptosis, and the just write the reason in the next part of it. The cells between fingers 2 and 3, you can see, have not undergone programmed cell death, right?
yeah,
Is it? So the tissue
they
has
have
linked fingers 2 and 3 during the development.
undergone like the permanent cell death.
Programmed cell death, bete. Kunain, programmed cell death. Apoptosis is programmed cell death.
Cell death. So that means that the tissue that's now
At
death.
certain point, the cells would die themselves, right? Because they are programmed to die, right? So the
Yeah.
process that might be regulatory by the EPHS7 gene is apoptosis that we can say. So the reason for this is the cells between fingers 2 and 3 have not undergone programmed cell death. So the tissue that linked fingers 2 and 3 during development has not been lost, right? Because if the programmed cell death is not occurred, so ultimately the tissues would keep on linking the fingers. That's here in this case 2 and 3. So they would not, uh, I would say, um, they would not lose during the development process. Are you getting?
Yeah.
Write down the answer, please.
Okay. Done.
Done?
Yeah.
Okay. This is what we have done. So uh, I'll send you another source from the same topic, right? So that's you
Mhm.
have to do and send me back for evaluation, right? So this is what we have covered, means from one source, and you just need to work on this another source from the same chapter. Okay, so this is
Okay.
what we have for today. I think let's conclude this for today, Kunain. Anything that you want to ask, you can please.
No, it's fine.
Okay, so let's conclude this for today. Take care of yourself. Bye bye. See you in next class, inshallah. Bye.
Okay, bye.